now we have Steve Macknik. I guess sight is a terrible thing to lose, right? And you're going to talk about restoration.
Yeah. Okay. Well, I bet when you all arrived here today, you weren't exactly sure why the title of the symposium contained the word resilience. But now you know. And I'm the only thing standing between you and your dinner, so that scares me. So I'm going to rush right along here. Now, this project is something I've started with a large group of people across multiple institutions to try and use what we know about the neuroscience of connectivity between the LGN and V1 to develop a new type of prosthetic that can actually restore vision at the highest attainable acuity. Okay, so current prosthetic technology actually will take a blind person and make them slightly less blind, but there's nothing close to a prosthetic that will restore vision to someone so that they're not blind. Okay, so meaning that they have better than 2400 vision. Okay, so let's look at this. So first off, the reason why we're doing this is because there is no therapy that restores high acuity foveal vision due to catastrophic damage, due to the damage to the eye, to the optic nerve, to the retinal ganglion cells. There's just no recourse at all. Over 200 million people suffer from foveal vision loss for one reason or another. There's many more diseases than this. It's about 3 to 5% of the world population. So how do we get at this? Well, first off, vision has two main premises you got to know. Susanna already alluded to this. If you hold out your thumb like this at arm's length, please do it. You will see that your thumbnail is in perfect 20/20 vision, but the only thing that is in 20/20 vision is your thumbnail. Everything else, the other 99.9% of your visual field, you are legally blind. Okay? So when you're trying to make a prosthetic for vision, you need to fix that thumbnail, and if you don't fix that, you're still going to be blind. And your prosthetic better deal with eye movements. Okay? So the other premise is if you're going to fix that, you need to understand that the answer to vision lies in the stars. So if you go outside and you look at the night sky and you look at the smallest, dimmest star in the sky, okay, do you know what the difference between that and the brightest star in the sky or the largest star in the sky? It's actually not size. They're exactly the same size as far as your vision's concerned. They're smaller than one photoreceptor. Every single star, no matter their size, is smaller than one photoreceptor in your eyeball. Okay? The reason one looks bigger than the other is there's so much light that it scatters and it causes photoreceptors around a given photoreceptor to light up and you see a larger object due to scatter. So prosthetic stars, if we could simulate and cause prosthetic dimmest stars in the sky, the smallest star in the sky, and then we could make patterns of those, we could actually recreate any object in the visual world that you can possibly see, because that's what your real visual system does. And that's how your real visual system does it. Okay? So here's how the real visual system works. During normal vision, the retina transduces light into neural impulses that travel through the optic nerve to the lateral geniculate nucleus of the thalamus, the LGN. These signals then travel further through the optic radiations into the primary visual cortex, V1. In the layer 4 LGN inputs to V1, where the retinotopic inputs are aligned in stripe patterns, on and off stripes are arranged perpendicularly to ocular dominant stripes, so that every single point in retinotopic space is represented by one on and one off input from the LGN from each eye in a concise module called the hypercolumn, shown here in green, which subtends about 1 square millimeter of V1 cortical surface. So every single thing about a given point in space is contained in that green square. Everything you've ever seen in that point in space. You've got left and right eye is the two halves, and you've got on versus off. On is the dimmest star in the sky, white on black, and the smallest period you can see on a white piece of paper is the smallest black thing you can see. Okay? You've got roughly 1,200 of these hypercolumns on each side of your brain, and that's everything you've ever seen. Okay? So what we have to do, and everything in this 1 millimeter square, each one of these quadrants, everything within one of these quadrants is fungible. If I stimulate a small spot in one part of one of those quadrants, and I move it to a different spot in the same quadrant, you'll see exactly the same thing. Nothing moves, nothing changes. If I make the spot twice as big, it doesn't get bigger, it gets twice as bright, okay? Because everything in that point in space is the same as far as we know. All right, so how does the brain achieve high acuity? Well, synaptic precision is critical because it avoids unwanted targets. That optimizes resolution, and that optimizes signal to noise. Prosthetics, therefore, also need synaptic resolution, and electrodes cannot achieve this. Electrodes' deficiencies include that electric fields cannot precisely select targets. All excitable membranes are affected by an electrode that's stimulating. You're going to hit unwanted neurons because of the size of the electric field, even from a very precise electrode such as the ones that Musk has developed for Neuralink. Okay? You're going to hit glia, you're going to hit blood vessels, all sorts of stuff you don't want to hit, and that's going to be a problem. You're going to get phosphenes instead of the smallest star in the sky. So optogenetics, though, has the precision of light. So if you take optogenetics and you cause an individual cell to have photoreceptors, proteins in it, you can now get for synaptic precision if you have the right kind of circuit and the circuit's laid out properly, like our hypercolumn is, where you have individual LGNs, groups of cells in a given layer so that you can hit those and not hit unwanted neighbors. Okay? So you can hit the targets of interest only without hitting anything you don't want to with synaptic precision because of the way the circuit's made combined with optogenetics. Okay? So this is how that would look like. To restore the visual pathway of a person who is blind, we must develop technologies to replace these functions. In our solution, the patient will wear a pair of eye-tracking glasses that monitors the center of their gaze on the visual scene. To get the information from the camera into the brain, we will first inject adeno-associated virus into the LGN, where it will deliver genetically encoded optogenetic light-sensitive proteins. These channel-rhodopsin proteins will be produced within the LGN cell bodies before percolating up through the optic radiations into the LGN boutons in layer 4 of V1. We can now optically stimulate those LGN boutons with light to generate precisely targeted and purely excitatory glutamatergic activation in V1 using the same synaptic mechanism used in normal vision. Optimal prosthetic function requires that we create a full feedback control loop that adjusts to variances in excitability of cortex. To read out V1, we will transfect a multicolored array of bioluminescent calcium indicators to stochastically barcode each cortical neuron with color. We can then read out activity in these neurons using Observe's camera, which is a similar device to your phone camera. It will serve as a hyperspectral photometer that detects colored photons emitted by the bioluminescent calcium activity in V1 neurons. Observe's coplanar imager emitter chip will have individual 25 micron square pixels in a 400 by 400 1 centimeter square array, each made up of an LED to stimulate LGN boutons surrounded by 5 monochromatic cameras with colored microfilters to optimize their differential chromatic sensitivity to the 5 transfected bioluminescent colors. Observe will be driven by cameras on the patient's spectacles, which will be spatiotemporal filtered to optimally activate V1 naturalistically. This signal is transmitted to Observe via a built-in ultra-wideband transceiver module inside Observe. We will then be able to read out the colored bioluminescent activity of V1 using the colorimetric spectrometers of the camera's pixel array. These spatiochromatic signals will be matched against a color lookup table corresponding to a high-resolution map of these same neurons. Okay, so that's how this project will work, and I want to thank especially the NSF, my partners, but also especially the NSF, who's been funding us for the last 5 years to develop the technologies involved here. And sorry we didn't have more time to talk about the details of the bioluminescence especially, but we have been getting excellent outcomes with both the optogenetic stimulation of the LGN inputs as well as the bioluminescent activity in V1 that we've been able to transduce. Anyway, thank you very much.